Welcome to this video, dear learners, and our topic for today, enzymes. So enzymes, this is quite an interesting topic in biology, and we are going to understand that enzymes are really important for various activities in the human body. Okay, so for those new to the channel, please hit the subscribe button, and if you haven't watched the previous videos, I would advise that you watch the previous videos, because most of the things we'll be discussing, some of them have already been discussed. So on enzymes, the definition for enzymes, this is like the simplest definition, okay, in biology. It's just a two-word definition for enzymes. When you're defining an enzyme, we define it as a biological catalyst. It is defined as a biological catalyst. That's the definition for an enzyme. Okay, it is simply a biological catalyst. Okay, so now we call them biological because they catalyze reactions inside the human body. Okay, so a catalyst, okay, we need to understand first what a catalyst is. Okay, so the word catalyst, okay, so a catalyst is any substance that can speed up the rate of a reaction. So, and what enzymes simply do is that they speed up, okay, the rate of chemical reactions. Okay, so enzymes are catalysts, okay, and catalysts, they speed up chemical reactions. Okay, a catalyst always speeds up a chemical reaction. So we have the definition here, biological catalyst, and we've defined what a catalyst is. So what enzymes simply do, they speed up chemical reactions in your body. Okay, there are a lot of chemical reactions that happen in the human body, and they have to be catalyzed by these enzymes. Okay, so enzymes, we can say enzymes, okay, enzymes catalyze reactions. Okay, enzymes catalyze reactions in the human body. Okay, enzymes catalyze reactions inside the human body. Okay, they catalyze these reactions. Okay, so whenever you have a reaction, let's say A plus B, okay, most of the reactions are reversible. C plus D. Okay, so this is a reaction. So in order for this reaction to speed up, okay, so actually you find that this reaction, okay, can take 5 hours. Okay, can take 5 hours to take place. Okay, but when you introduce an enzyme here, you find that the reaction takes only 2 hours. Okay, so you can see from the 5 hours, with an addition of an enzyme, it only take about 2 hours to finish the reaction. Okay, which is actually a good thing. Okay, so enzymes, so there are some reactions actually that are not even going to take place when the enzyme is missing. Okay, when the enzyme is missing. Okay, so some reactions will not take place if the enzyme is missing. Okay, so you can see how important these enzymes are. You can see their importance, okay? Just how important the enzymes really are. Okay, so saying catalyst speeds up a chemical reaction. Okay, so now what happens is this, in any reaction you're going to have two things. Okay, usually you're going to have an enzyme. Okay, so you're going to have an enzyme. Say this enzyme has a shape like this. Okay, this is your enzyme. Then you have a substrate. Substrate has a shape like this. Okay, then now these two will have to react together. Okay, they have to react together, and then what you're going to get, most of the reactions said they're reversible, the human body. Okay, so now what you're going to have is something like this. Okay, and then then you're going to have something like this. Okay, so now this here is an enzyme, and then this here is the substrate. Sorry, it's no longer a substrate now, it's a product. Okay, it's a product. Then here you've got the enzyme substrate complex. Okay, enzyme substrate what complex. Okay, so now this is what is happening here. So the enzyme binds. Okay, so here they bind together. So before anything can happen, this enzyme has to bind to the substrate. Okay, these two, they have to bind, and when they bind they form an enzyme substrate what complex. Okay, they bind to form, okay, they bind to form an enzyme substrate complex. Okay, then at the end you have the enzyme. So the enzyme here, it remains unchanged. Okay, the enzyme here remains unchanged. Okay, it remains unchanged, it does not change. But you can see the substrate here has changed to be a product. Okay, so this is what we call catabolism. Okay, such type of reactions we call them catabolism. Why why are they called catabolism? Because they involve breaking down. So this substrate has been broken down into two substrates. So these such type we call these catabolism. Okay, or catabolic reactions. Okay, catabolic reactions. So we are going to say enzymes catabolize, or we can say catalyze both, they catalyze both anabolic and catabolic reactions. Okay, they catalyze both anabolic and catabolic reactions. Okay, so what I want you also to know is this. So this is your enzyme. So this is not how enzymes really look like. They have different shapes. So this is the shape of your enzyme. Okay, this is the shape of your enzyme. So this site here where the substrate binds is called the active site. Okay, active site is where substrate binds. Okay, the active site is where the substrate binds. Then these other sites, they're known as allosteric sites. Okay, allosteric sites. Okay, so the active site is where the substrate does what? Binds, okay, to make that attachment, okay, to make that attachment. Okay, so this is how actually this reaction. So for any reaction to take place, the substrate must be bound. It must be bound to the active site on the enzyme. Okay, it must be bound to the active site of the enzyme. Okay, so always it has to bind to the active site. Okay, maybe write this one here. Okay, so for a reaction to take place, okay, the substrate must be bound to the active site, of the enzyme. Okay, it must be bound to the active site of the enzyme. Okay, just to bind to the active site of the enzyme. So if it doesn't bind, meaning the reaction won't be able to do what? Take place. Okay, if there's no binding there, the reaction will not take place. Okay, they have to bind together. Okay, so now that we've looked at how these enzymes happening, okay, let's look at some of the characteristics of enzymes. Okay, characteristics of enzymes. So under characteristics of enzymes, okay, so here be careful of these things. They'll appear in your GCE exams, even at grade 12. Characteristics of enzymes. So characteristics of enzymes, okay. So number one is that enzymes are proteins. Okay, a lot of them are proteins in nature. Enzymes are protein in nature. Okay. Number two, we've already said they are biological catalysts. Okay, so they are catalysts. Okay, meaning they speed up chemical reactions. Okay, they catalyze both forward, like I showed you. They catalyze both forward and backward, both forward and backward reactions. Okay, then they are specific in nature. Okay, here you are going to look at the lock and key theory. So enzymes are specific in nature. Okay, they are specific. Here we simply mean they're specific in that if you have your enzyme like this, I've already demonstrated this. So you have this substrate and this substrate. Okay. So between these two, okay, I'll call this one A, this one B. Okay. So what's going to happen is that for the reaction here to go on, for this reaction to go on, the first one A, okay, A is not going to happen. Okay, okay, it does not fit. Okay, does not fit. So this one here, it's not going to fit into the active site. Remember, we said this is the active site. Okay, the active site. But for the other one, the reaction would definitely take place because of the shape. Okay, because of the shape. So it's simply going to fit in. Okay, so B fits into substrate. Okay, B will fit into the substrate because of its shape. Okay, so we're going to say shape of substrate must match the shape of active site, of the active site. Okay, that's what we mean when we say they are what? They are specific in nature. Okay, they are specific in nature. The shape of the substrate must match the shape of the active site. Okay, it must match the shape of the active site. Okay, so if you have something like this is your enzyme. Okay, such a shape. Okay, here no reaction. Okay, if you have something like this, okay, here reaction. Okay, so here match, there's a match between the active site and the shape of the enzyme. Okay, so that's what we mean when we say enzymes are actually specific in what? In nature. Okay, they're actually specific in nature. So another characteristic we say they are affected by the pH. Okay, so this pH that we're talking about is simply the measure. Okay, pH is simply measure of alkaline. Okay, pH is the measure of acidity or alkalinity. Okay, the measure of acidity or alkalinity. So we have the pH scale. Okay, this we call it the pH scale. It has numbers starting from 0 up to 7, then 7 there up to 14. Okay, that's the pH. Okay, so this part below 7 is acid, above 7 is alkaline. Okay, so this side here it is acidic, this side here it's alkaline. Okay, so we're going to say pH of 2 is acidic, then pH of 3 pH of 8 is alkaline. Okay, it's alkaline. So now how does this apply to enzymes? Okay, so some enzymes, okay, some enzymes work well at acidic pH, while others work well at alkaline pH. Some enzymes work well at acidic pH, while others work well at alkaline pH. Okay, so for example, okay, e.g. at pH of 1 here, you can have an enzymes, e.g. an enzyme such as pepsin, okay, pepsin in the stomach, okay. Then you can have a pH, let's say, of 8. You can have an enzyme such as lipase from the duodenum. So these two enzymes, they have a specific pH at which they are going to work very well, okay. They have a specific pH at which they are going to work very well, okay. So that's how the pH really affects the enzyme, right? Some will work at acidic pH. For example, the stomach is acidic, okay. The duodenum is alkaline, so they have their own enzymes that will operate at that pH, okay. So let's look at another thing. Okay, so let's look at temperature. Okay, we can say enzymes are affected enzymes are affected by temperature, okay. Enzymes are affected by temperature, okay. They're affected by what? The temperature. So for this, I'll show it like this. Okay, so here I'm going to have activity of enzyme, then here the temperature in degrees Celsius, okay. So now the temperature is increasing this side, okay. Let's say here the temperature here it's 40 degrees, here let's say it's 37 degrees, okay. Here the activity of the enzyme is also increasing, okay. So find that here the temperature here is 0, the activity is low, okay. But as you go this side, you're increasing, okay. The activity also starts to go up till it reaches somewhere there, okay. Then here it begins to come down, okay. It's to come down like that, okay. So you're going to find that the activity of the enzyme was increasing and then it drops, okay, due to temperature, okay. So here the temperature here is low, okay. Here the temperature is high, okay. This is high temperature. Here the temperature is highest, okay. So at this type here, let's say this is the activity at A. You've seen the activity of the enzyme is what? It's low, okay. Let's say it's somewhere 5. As you increase here, it's 45 at the highest here. So here you find that here the activity of the enzyme is highest, okay. So at this temperature where the activity of the enzyme is highest, we call this the optimum what? Temperature, okay, where the activity is highest, okay. So here the activity is highest, enzymes are working at their best, okay. Enzymes are working at their best, okay. So this is the optimum temperature, okay. So at this temperature here, you can say enzymes are denatured by very high, okay, by very high temperatures. Enzymes are denatured by very high temperatures, okay. They are denatured by very high temperatures, okay. So to be denatured, they simply lose their shape, okay. So you can say they lose their shape. And once an enzyme loses its shape, the substrate won't be able to bind, okay. Remember I said the shape is important, okay. The shape is important. So here high temperatures, wonder why the activity here now at D is low, okay. Activity is low, okay. Here at B, the activity is at its best. Okay, here activity is at its best. Okay, at B. So we call this as optimum temperature. We call that optimum temperature. Okay, optimum temperature. So optimum temperature okay, we're going to say enzyme activity is working at its best at the optimum temperature. Okay, the activity of the enzyme is at its best. Okay, it's at its best. Okay, so also going to say another characteristics. The rate of reaction of an enzyme is dependent on the concentration of the substrate. Okay, so we are going to say the higher the concentration, of the substrate, the higher the activity of the enzyme. The higher the concentration, okay, the higher the concentration of the substrate, the higher the activity of the enzyme. Okay, the higher the activity of the enzyme. Okay, so looked at how many? 1, 2, 3, 4, 5, 6. Okay, 7 characteristics of what enzymes. Okay, so let's just have an example of enzymes. Okay, so enzymes okay, and carbohydrates. Sorry, my ink it's not that clear. Okay, so enzymes under carbohydrates, we have carbohydrate enzymes. These are enzymes that break down carbohydrates. Enzymes that break down carbohydrates. Okay, so the type of carbohydrates we looked at polysaccharides, disaccharides, and monosaccharides. So you have starch, which is a polysaccharide. The other name for starch is amylose. Okay, you have maltose, you have sucrose. Okay, you have lactose. Okay, you have lactose. Okay, so here now the enzymes that will break down these things. Okay, if you want to know which enzyme will break down, so just change the last 3 words. Okay, you put ASE. Okay, ASE. So for the first one, amylose, it will be broken down by amylase. Maltose will be broken down by maltase. Sucrose will be broken down by sucrase. Lactose will be broken down by lactase. Okay, by lactase. Okay, by lactase. So these are enzymes. Okay, or these are enzymes. Okay, so amylase breaks down amylose, which is starch. So if I ask you what enzyme breaks down starch, it's amylase. What enzyme breaks down maltose is maltase. What enzyme breaks down sucrose is sucrase. What enzyme breaks down lactose, it is lactase. Okay, it is lactase. Okay, then what about lipids? You're going to do the same thing, change the last two, put ASE. Okay, so the enzyme is lipase. Okay, then for proteins, okay, for proteins you have got peptides. You remember we talked about peptides in our previous video under nutritions. Peptides are broken down by peptidase. Okay, peptidase. Okay, they're broken down by peptidase. Okay, so just an example of enzymes, and we'll look at them in detail when we are going to discuss about digestion. Okay, so look out for the next topic. We're going to start will be digestion, and then we'll go to photosynthesis. Okay, so what we've covered in this video are enzymes. Okay, we've said enzymes are simply biological catalysts, and a catalyst is any substance that speeds up the rate of a chemical reaction. They make the reaction fast. Okay, so as demonstrated here, if a reaction was supposed to take 5 hours, with the addition of an enzyme, it can just take about 2 hours. Okay, so some reactions will not take place without the enzyme. So this has been understood well, even in medical field. There are a lot of diseases that can come about simply because someone is lacking an enzyme. Okay, so we talked about the catalyze both anabolic and catabolic reactions. Okay, and the active site, this is where the substrate binds. Okay, then from here we went on to discuss the characteristics of enzymes. They are proteins in nature. They are catalysts. They catalyze both forward and backward reactions. They are specific in nature. Okay, the substrate must bind to the active site, just like a lock and a key. Otherwise called the lock and key theory. Just like a lock, the key fits inside the lock. Okay, a wrong key is not going to fit, and here a substrate with a wrong shape is not going to fit in the active site of the enzyme. So they're affected by pH. Some they work well at acidic pH, some they work well at alkaline pH. Okay, talked of temperature. So you increase the temperature, the activity of the enzyme is increasing. But if you go beyond the optimum temperature, the activity goes down, and this is, we said, the enzymes are denatured when the temperature is very high. Okay, so the activity of the enzyme is working best at optimum temperature. Then you said the higher the concentration of the substrate, the higher the activity of the enzyme. We've given here just some examples of these enzymes under carbohydrates in the digestive system. Okay, so the food we eat has to be broken down when digestion is taking place, and this is helped by these enzymes: amylase, maltase, sucrase, lactase, lipase, peptidase, and we'll look at many more of these enzymes when we discuss digestion. Okay, so just stay tuned, click the subscribe button together with the like button, like and share this video to your friends, and also keep on following our channel for more videos, and just to understand how everything is happening in the human body in biology.